Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is the vernalization. Okay, this is related to the plant physiology, and what is first we have to know the vernalization process. Plants have evolved the ability to alter their developmental program in response to environmental stimuli. So we all know that we are in adaptive condition. That means we have to always accustom with the environment. So for our growth and obviously for our existence. So in case of plants, they have to adjust obviously with the environment, and that's why it is saying that their developmental program in response to environmental stimuli. That means their development generally related to with the environmental stimuli. So what two important environmental stimuli that are, first one is that temperature, and the second one is that photo period. Okay, temperature and photo period. And the major switch in the developmental program in the transition to flowering. This is very important. Sorry if some noises are hearing, so you just ignore that. Okay, it is not from my room, that is from the outside. It is coming from outside. So what is saying that major switch in the developmental program in the transition to flowering. So it's in case of plants, the flowering is very important, and this flowering process is related with the environmental factors. Okay. In many plant species, the timing of this transition is determined by the seasonal changes. So transition process, that is transition to flowering process, that is determined by the seasonal changes. So season is very important, and we have seen that some cases some flowers they can grow in winter season, and some flowers they can grow in the summer season. So this may be varied in different cases. Okay. So here the two important factors are what? One is the photo period, another is the temperature. These are the two factors that plants monitor to determine the correct time to flower. So in the two factors which are very responsible for flowering, and plant obviously they can sense that the two factors and they adjust themselves when which time is favorable for them to for flowering. Okay. Vernalization, I have written here the short form of the vernalization is this is the VN. This describes the promotion of flowering after exposure to cold 0 to 5 degrees Celsius. So vernalization is what? This describes the promotion of flowering after exposure to 0 to 5 degrees Celsius. That means it is necessary the plant for getting flowering 0 to 5 degrees Celsius exposure is needed to cold temperature, that means cold condition. This vernalization results in the acquisition of competence to flower. That means after vernalization, plants do not necessarily initiate flowering but acquire the competence to do so. That means this exposure to cold temperature, it is not initiate them to become to adjust the flowering. No, it just help them to mature their body or to create some changes in their body to for flowering. Okay. After vernalization, plants do not necessarily initiate. Okay, I have said that. It is reversible and can be lost as a result of exposure to de-vernalization. So this helps in another way in the flowering process to continue within the plant's body, or create some changes in the plant's body to for flowering. But in case of de-vernalization is what? De-vernalization is that high temperature or its reversible condition. So it's 0 to 5 degrees Celsius It is the cold temperature or cold condition. This is called the vernalization. But its reversible condition is the high temperature. And if the plant is exposed to temperature, that means their flowering process becomes stopped. Clear? So here two mechanism is very importantly explained by the scientist. One is the hormonal theory and another is the genetic theory. In case of hormonal theory, these are the generally vernalized plant. Example is that henbane. Okay, this is. But this theory, this is applicable for non-vernalized plant. So what is saying that this studies involving the grafting and the localized cooling and have shown that the apical meristem is the site of cold perception during the vernalization, and that vernalization causes the meristem to become competent to flower. So what is saying that grafting and localized cooling is that experiment, and what is the observation and conclusion is that cold temperature that creates meristematic tissue to induce or meristematic tissue to induce to divide, where at the apical meristem area, at the apical region, you can say, at the apical region, and that's why the cell division occur in that region, and as a result the plant's body become competent to flower, or flowers there a chance of flowering. That situation is created. Thus dividing cells are a prerequisite for vernalization, and after grafting experiment it was seen the existence of vernalin. Now this term vernalin is very important. So this vernalization process that induce the vernalin is one kind of hormone. Okay, not vernalin. Vernalin is one kind of hormone that becomes stimulated due to this vernalization process, and as a result the plants become mature or competent for the flowering process. Clear? So this is the ray diagram, and cold temperature that stimulate the vernalin and that stimulate the plants. Okay, here the flow region is one kind of gene. Okay, and this become activated due to the stimulation of the or activation of the vernalin hormone. That flow region gene that becomes activated, and which is responsible for flowering. Clear? This is the hormonal theory. Another important theory is the genetic theory. This saying that vernalization involves the epigenetic changes in the expression of gene FLC. Okay, epigenetic changes of which gene? FLC gene. FLC gene is flowering locus C gene. This FLC that encodes a MADS box. MADS, the full form is that minichrome agamous deficiency serum. Okay, MADS box. This transcription factor, and that represses the flowering. So what is saying that FLC encode MADS box transcription factor, and that represents the flowering process. Clear? And it is expressed predominantly in mitotically active regions. Mitotically active region means the region where the cell division is occurring. Now, in Arabidopsis, FLC works by directly repressing the expression of the FT. FT means flowering locus T, FT gene. So it represses, FLC directly represses which gene? FT gene, and obviously SOC1 gene. The full form is SOC1 what? The suppression of overexpression of constans 1, and FD. FD means flowering locus D genes. So these at the shoot apical meristem. So due to the expression of this FLC gene, that deactive or suppressed the activity of these genes, which are that one is the FT gene, second is the SOC1 gene, and third is the FT gene. So these genes become deactivated and that's why the flowering process is not activated due to the activation of the FLC gene. Clear? That means FLC represses that genes, and that's why the flowering process is remain in stopped condition. In response to VN means vernalization or cold temperature, FLC mRNA and protein reduced. So activations of these FLC genes that become reduced, and the reduction in the FLC expression that involves the chromatin remodeling of FLC, and that requires VIN3. So it is another important protein. This name is that vernalization insensitive 3. This becomes activated, and the product of other genes, that is VRN1 and VRN2, that responsible for the expression of this FLC gene, but in different manner. That means another way. And its products are not for FLC silencing. So what is saying that? That means the FLC mRNA, when the protein reduced, okay, when the proteins that is produced from the FLC due to the vernalization, when this reduced, that means the that genes become activated, which are already suppressed by the FLC gene. So flowering, that will be possible. Clear? So what is saying that cold temperature, that means VIN3 and VRA produced, and as a result PRC2 and methyl group, that all added with the histones with FLC gene. And this, what is the result of this? Shutting down of FLC gene and FT SOC1 that activated, and that means flowering possible. So here important thing that is played by the which VIN3 important role is played by this, because when the cold temperature exposure the plants to the cold temperature, that creates the important proteins, that is the VIN3. This protein is very important, produced, and as this protein produced, the PRC2, that is another protein, and methyl group all added with the FLC genes histone proteins. Clear? And as this all things are happening, that means the FLC genes expressions become stopped. Okay, so other genes that are helpful for flowering, that becomes activated, and the flowering process will be continued. Clear? So here PRC2, the full form is that, one second. So PRC2, the full form is that polycomb repressive complex 2. Clear? So this is the process vernalization I have explained here, and I think you understood. Okay, so if you are the newcomers and this topic is understood by you properly, then obviously share and subscribe this channel, and after subscription you just hit the bell icon for getting all the notifications. And I generally upload my videos at Saturday, Sunday, and sometimes Tuesday and Thursday, so you just check out. Okay, so thank you.